Hi everyone. Today we're going to be talking about bacterial immunity, specifically restriction enzymes and CRISPRs. In our presentation, we're going to start out with some background information, then we're going to specifically move into restriction enzymes, talk about what they are, talk about the bacteriophages, and some research and history. Then we're going to get specific within restriction enzymes and talk about the CRISPR-Cas systems, give an overview of what that is, specific mechanisms, and talk about some additional information.
In the background, it's clear that both restriction enzymes and the CRISPR-Cas system function as a prokaryotic immune system. This provides resistance to exogenous genetic materials, for example, plasmids or phages. This also acts as an acquired immunity for either the bacteria or the archaea. CRISPR allows microbes to distinguish foreign DNA from outside the cell from self-DNA from inside the cell. Exogenous DNA can be transferred through transduction, conjugation, and transformation. In this picture, you can see the different types of transfer for exogenous DNA: transduction, transformation, and conjugation. In transduction, it's evident that the DNA is being transferred from one bacterium to another by a virus. The foreign DNA is introduced via a viral vector. The DNA that's being transferred is the phage DNA and the cell DNA. Transformation is shown to be the genetic alteration of a cell resulting from direct uptake, or incorporation and expression of exogenous genetic material through a plasmid. Lastly, conjugation occurs when genetic material is transferred between bacterial cells by direct contact between the two cells via the pili, which you can see touching in the picture. Microbes also use other strategies in order to avoid contracting exogenous DNA. This is in addition to their immune system that's innate. Some of these include prevention of absorption, blocking of injection, and abortive infection.
Now let's talk more specifically about restriction enzymes. Some background history about restriction enzymes. It all started when Swiss microbiologist Werner Arber and his assistant Stuart Lind proposed that there were specific sites and sequence on the genomes where the restriction enzyme activities were occurring. This was called endonucleatic scissions, also known as endonuclease R, later named and changed to ECO-B. Then, after this, other scientists discovered ECO-K. Shortly after that, Hamilton, Smith, and Kent Wilcox, they isolated and characterized the first restriction enzyme from second bacterial species known as Haemophilus influenzae. They discovered that the restriction enzyme HIN2, which degrades foreign phage DNA, but it did not I gotta read that over. Hamilton, Smith, and Kent Wilcox discovered that the restriction enzyme HIN2 degrades foreign phage DNA, but not the bacterial host DNA. Then after this, Smith and Thomas Kelly identified nucleotides, the actual nucleotide sequence of the specific site where HIN2 cleavages, also known as the recognition and restoration site. Talking more generally about restriction enzymes, there's 800 different restriction enzymes from various bacteria, and they recognize over 100 restriction sites. Specifically, there are 3 different types of restriction enzymes. Type 1, which recognizes and binds specific DNA sequences, but cleaves non-specifically ECO-B and ECO-K. Type 2 recognizes and cuts within the recognition site and always cuts within the palindrome, while type 3 recognizes sequences but cuts at a different specific location, which is usually 25 base pairs away. Now let's take a look at the restriction enzyme cutting sites. Here we have an example of a type 2 restriction enzyme which cuts at a palindromic sequence. An example of this is EcoR1. As you can see, the top strand reads GAATTC. When you look at the palindromic side, the palindromic side will sequence forward and reverse on different strands. So the C will become the first letter, the T will then become the second letter, and then so on and so on. So the second strand will then read CTTAAG. In the second picture, we see like the whole cut that has been made. Moving on to the next slide, we see the restriction enzyme actually in action. In this picture, we have a 3D crystal structure of the restriction enzyme cutting the palindromic DNA sequence. In the top right, you have the red and yellow, which symbolizes one part of the restriction enzyme protein. And then in the bottom left, you have the dark blue and green, which is the other half of the protein or the restriction enzyme. In the middle, you have the DNA that is going to be cut. One part of the restriction enzyme will cut the one strand, and the other part of the restriction enzyme will then cut the other strand. Some further research that we have about restriction enzymes is that they will only cut sequences that are unmethylated. If they are methylated, the sequence is protected and will not be cut. Also, phages that have already survived replication in a bacteria are protected from being cleaved by restriction enzymes in another bacteria of the same strain. So, getting to the point of all this, why do we want to use restriction enzymes? Well, we want to use restriction enzymes because it's used to manipulate specific segments of the DNA, recombining DNA molecules from different genomes to identify and study genes and gene expression. And almost all molecular biology benchwork begins with the use of restriction enzymes.
So what exactly are restriction enzymes? I have included this quotation in the presentation strictly for the fact that I find this to be a near perfect explanation of what restriction enzymes actually are. I'll read it aloud. When I come to the laboratory of my father, I usually see some plates lying on the tables. These plates contain colonies of bacteria. These colonies remind me of a city with many inhabitants. In each bacterium there is a king. He is very long but skinny. The king has many servants. These are thick and short, almost like balls. My father calls the king DNA and the servants enzymes. My father has discovered a servant who serves as a pair of scissors. If a foreign king invades a bacterium, this servant can cut him in small fragments, but he does not do any harm to his own king. Now, it's definitely worth pointing out that this is made by a 10-year-old who is the daughter of Werner Arber, who was mentioned earlier by Saad. So, in more sophisticated terms, a restriction enzyme is an enzyme produced by specific bacteria that serves the function of cleaving DNA molecules at a particular sequence of bases. All restriction enzymes protect bacteria against invading viruses. We will take a look about how this works a little bit later. Now I'm going to give you a little history of restriction enzymes. Well, as earlier mentioned, they were discovered by Swiss microbiologist Werner Arber with the help of graduate student Stuart Lin. He discovered these while researching what they termed host-controlled immunity, which later became known as a restriction of bacteriophages. Before we go any further, I'd like to explain what bacteriophages are and what they do. So, what are bacteriophages? Bacteriophages are, in simplest terms, viruses that infect and replicate within bacteria. Alright, so there was prior research conducted by Salvador Loria and graduate student Mary Human, along with many others. In their research, they found that bacteriophages were host-specific. When I say host-specific, I don't mean that bacteriophages can infect one and only one type of bacteria. It is just that each phage survives and flourishes in certain strains of bacteria more than others. Now, Arber was curious about the host specificity and wanted to know why some were restricted or immune and others were not. Now this is a simple diagram just showing the bacteriophage structure, and it's pretty much self-explanatory with the head that contains the DNA and then the sheath along with the protein and the tail fibers that actually feel around the bacteria to find the exact site where it would like to bind to before it injects its DNA. And then the spikes on the bottom that you see actually hold it in place. Now this is a picture of many phages bound to the membrane of E. coli. This is obviously a manufactured photo after exposing the E. coli bacterium to excessive amounts of phages. However, this could happen if a neighboring cell lyses, which was previously infected. Now this is a diagram of restriction enzyme mechanism. The DNA methyltransferase, also known as eco-RV methylase in this case, which is that purple spiky ball-looking thing, adds methyl molecules, CH3, to nucleotides within the bacteria's genome. Bacteriophages inject the DNA into the cell, and the eco-RV nuclease, which is the restriction enzyme, cleaves unmethylated viral DNA at a specific or non-specific location.
Now we can talk a little bit more specifically about a type of restriction enzyme, the CRISPR-Cas system. In essence, CRISPR stands for clustered regularly interspaced short palindromic repeats. Keep this in mind for the picture that you'll see in an upcoming slide. CRISPR loci consist of several non-contagious exogenous DNA repeats, which are then separated by spacers. Specifically, spacers are put in between CRISPR repeats, and CRISPR spacers are a mugshot of the foreign DNA. This allows the immune system to recognize and silence exogenous genetic material. Here's a picture of a CRISPR mechanism. As you can see, the CRISPR precedes the spacer. With that, the different Cas or CRISPR-associated genes can go and cut and silence different strands of DNA which are held in the mugshots of the spacers. Some additional facts are that the CRISPR-Cas system is found in approximately 90% of archaeal and 40% of bacterial genomes. They range in size from 24 to 48 base pairs. As Saad mentioned earlier, restriction enzymes show palindromic symmetry or dyad symmetry. Evidence for functional diversity amongst CRISPR subtypes has been shown through research of different kinds of bacteria and archaea. CRISPRs are typically located on the chromosome, but some have been identified on plasmids. Mostly, though, they remain in the chromosome. Spacer addition can provide phage resistance, meaning that it's a type of immunity for the bacteria or archaea. The CRISPR system is made up of a bunch of different Cas genes, or CRISPR-associated genes. For example, Cas1 is a double-stranded DNA or dsDNA endonuclease. An endonuclease is something that cleaves the bond within a polynucleotide chain. This is involved in the immunization process. Then, Cas2 acts as a sequence-specific endoribonuclease, which cleaves single-stranded RNAs, or ssRNAs. Lastly, CRISPR repeats become available to the Cas system through transcription. The mechanism can then be seen through the exogenous DNA being processed by proteins encoded by Cas genes. These processed elements are inserted into the CRISPR locus, or a specific location of a gene or DNA sequence on a chromosome. Then, RNA from the CRISPR is processed by Cas proteins into sgRNA, or small guide RNA. Each RNA is composed of individual exogenous materials. Lastly, the sgRNAs guide Cas proteins to identify and silence exogenous material when seen again. Cas genes encode a large and heterogeneous family of proteins. This includes functions similar to nucleases, which cleave phosphodiester bonds between nucleotide subunits, helicases, which unpackage an organism's genes, polymerases, which synthesizes polymers of nucleic acids, and polynucleotide bonding proteins. Most CRISPR repeats are partially palindromic, which Saad referred to earlier. In addition, CRISPR has the ability to undergo horizontal transfer between genomes. This horizontal transfer can be mediated by plasmids, megaplasmids, and prophages. This is a picture of the horizontal transfer that can occur, similar to the slide in our background information, which explains the way that exogenous DNA can transfer from different cell to cell.